Very good morning, students. This lecture is citric acid cycle, and citric acid cycle is nothing but Krebs cycle, and the end product of pyruvate dehydrogenase complex, okay, that is acetyl coenzyme A. This is the end product of pyruvate dehydrogenase complex. Is going to react with oxaloacetate, the 4-carbon compound, and the 4 plus 2 carbon going to form a 6 carbon molecule that is called as citrate. So that is why it is called as citric acid cycle. So we have a sequence of biochemical reactions, and it is a cyclic reaction because again this oxaloacetate is going to be formed, and again it will receive another acetyl coenzyme A. This is a cyclic pathway. So here the first reaction, citrate synthase, involved in Chazen condensation process. The methyl group of acetyl coenzyme A converted to methylene in citrate. Okay, the methyl group of this one is converted into and methylene in citrate. So this CH3CO moiety attached with going to be attached with this CO moiety, and so this CH2COO and CCOO and the CH2CO will be formed. Okay, this is called a citrate. Just you can attach this one to this carbon, and this oxygen is converted into hydroxyl moiety, okay, by addition of hydrogen donated by water molecule, and this coenzyme A is liberated. So this coenzyme A liberated as a coenzyme A molecule COSH, and the product is citrate. So that is why citrate synthase is the name of the enzyme. Second reaction, there are two second reaction, that is by the enzyme aconitase. So 2A, that is dehydration or rehydration. Hydroxyl moiety of group of citrate repositioned in isocitrate to set up decarboxylation in the next step. So an intermediate is formed, okay, that is called as cis aconitate. So this cis aconitate is the formation of an intermediate molecule. This cis aconitate is intermediate. That means it is not a stable molecule, and this stable molecule further rehydrated into again stable molecule isocitrate. Actually, aconitase is involved in the formation of citrate to isocitrate, and this is an intermediate. That is why it is given in the bracket. Okay, and so this is liberation of water and absorption of water also. That is dehydration and rehydration. And CH2COO and CHCOO and CHOHCOO. So this is called as isocitrate, and so this isocitrate further involved in the another reaction, that is dehydrogenation. So usually the dehydrogenation involves that is removal of hydrogen molecules, and this removal of hydrogen molecules will be converted into NADH plus H plus. So this NADH formation is here. That is a very important role. So this energy rich molecule, this will go directly into the electron transport chain. So this reaction, that is first reaction, second reaction, and this is the third reaction. Oxidative decarboxylation, this is called as oxidative decarboxylation. And the OH group oxidized to carbonyl group, okay, which in turn facilitates the decarboxylation of stabilizing carbon ion formed on adjacent carbon. And this carbon dioxide is also liberated. So we are exhaling the carbon dioxide. That carbon dioxide is coming from Krebs cycle. So this is the waste product, and this has to be removed from the system by the respiratory system, okay, through the blood circulation. And this isocitrate dehydrogenase further forms alpha ketoglutarate after the removal of, okay, the NADH plus H plus, and the end product is the byproduct of this reaction is alpha ketoglutarate. Alpha ketoglutarate. Okay, the keto moiety is in the alpha position. That is why alpha keto glutaric acid. So this alpha ketoglutarate again undergo dehydrogenation. So this is also oxidative decarboxylation. So here also two maybe the hydrogen molecules are liberated, and these two liberated in the form of NADH plus H plus. Again, this will go into the electron transport chain, and here also one carbon dioxide is liberated, and coenzyme A is going to be attached. So alpha ketoglutarate to succinyl CoA. Succinyl moiety with coenzyme A. This is coenzyme A and attached with the succinyl moiety. Okay, succinyl means nothing but it is succinic acid and succinyl CoA. Coenzyme A going to be attached, and this coenzyme A going to be removed in the next reaction. So this alpha ketoglutarate dehydrogenase complex. Complex means it is a multi-enzyme complex, and this will liberate succinyl coenzyme A. This succinyl coenzyme A further formed into succinate by the removal of coenzyme A. So this coenzyme A is going to be removed by the next enzyme succinyl CoA synthetase, and GDP plus PI. GDP is equal to 1 molecule of ADP and GTP is equal to 1 molecule of ATP. So that is why it is given the brackets. So GDP plus PI converted to GTP plus PI, and this is called a substrate level phosphorylation. That means it's very, very important. Oxidative decarboxylation is also very important, and substrate level phosphorylation, that is energy of thioester bond conserved in the phospho anhydride bond of GTP or ATP. Okay, so this energy, the liberation of CoA, coenzyme A, is saved in the form of GDP2, GDP plus PI2 and GTP. So this phosphate, inorganic phosphate moiety attached with GDP and to be formed in the form of GTP, this energy rich molecule. So this one GTP is equal to one ATP. That is why it is given either GTP or ATP. Okay, this is called a substrate level phosphorylation. The substrate liberates, that is a breakdown of a thioester bond that liberate energy. That amount of energy is utilized for the fusion of the phosphate with GDP, and the GDP is converted into GTP. So this PA is nothing but inorganic phosphate. The succinate is a product now. Succinate again undergo dehydrogenation, and this dehydrogenation means removal of again two hydrogens. So this two hydrogens are removed not in the form of now NAD. So this is in the form of FAD to FADH2, and FAD is the incoming molecule, and maybe FADH2 is formed, and FADH2 receives two hydrogens. Now the product is called as fumarate. So from here, two hydrogens are removed, here one hydrogen and here one hydrogen. So this is CH2 converted to CH, okay, and FAD is converted to FADH2, okay. And next one is, in the next enzyme is fumarase enzyme, that is seventh enzyme is fumarase, and the fumarase utilizes water molecule. This is a reversible reaction, and this one forms malate. So fumarase utilizes water molecule. Water molecule consists of H and OH. Just you can see here, okay, CH and CH is converted into CH2, CH and OH. The hydrogen is given here, one hydrogen. That is why CH is converted to CH2, and another hydroxyl given here, the second, that is CO, CH, OH. So this is malate formation, fumarate to malate, and malate to oxaloacetate by the malate dehydrogenase complex. So this malate dehydrogenase complex again utilizes NAD, NAD to NADH plus H plus. So that is why it liberates one NADH plus H plus. So the product is oxaloacetate, and this oxaloacetate is further formed into the combining with acyl coenzyme A and forms citrate. So oxaloacetate combined with acyl coenzyme A forms citrate. So that is another cycle of reactions. So with this we can conclude citric acid cycle, and we can go to the regulation of citric acid cycle. Yeah, this is the regulation of citric acid cycle. You can see here this is the rate limiting step, and this is unidirectional reaction, and this is reversible, reversible, unidirectional, unidirectional, reversible, 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 and reversible. So most of the unidirectional reactions are controlled by regulatory enzymes. Okay, you can see here pyruvate dehydrogenase complex is already, we already completed, okay, that one. So coming to the TCA cycle components, number one, citrate synthase is the first enzyme is a regulatory enzyme. and isocitrate dehydrogenase is a regulated enzyme, and alpha ketoglutarate is a regulated enzyme. So already three enzymes are involved in the regulation of the fine tuning of TC cycle, and we can see one by one citrate synthase, and the green color indicates that is positive modulator, and this red color indicates negative modulator. Positive modulator is ADP and negative modulator is NADH, succinyl CoA, citrate, and ATP. You can see here why this ATP, citrate, and succinyl CoA NADH is inhibiting this enzyme means ATP is energy rich status. If ATP is there, there is no need to produce NADH. Citrate, that shows okay this energy rich status. If it is also there, if more citrate is there, there is a byproduct, an end product of this one. If more citrate is there, no need to synthesize the citrate, and more succinyl CoA is also there. That means more the products are formed. That is why this enzyme is inhibited. NADH is also more means that is not automatically okay, that will be inhibited. ADP is inducing, or otherwise the positive modulator. So ADP, why ADP is inducing means ADP that shows energy less status, that is energy deficient status. So that is why ADP will induce this enzyme, citrate synthase. And second regulated enzyme is isocitrate dehydrogenase. This isocitrate dehydrogenase may be controlled by a regulated enzyme controlled by two factors. One is positive modulator and this is negative modulator. Negative modulator as such, okay, this ATP same, and CA2 plus and ADP. So this ADP is also same, both. And next one is alpha ketoglutarate dehydrogenase complex. The positive modulator is CA2 plus, like this one, and succinyl CoA and NADH. Succinyl CoA and NADH is the same. Succinyl CoA and NADH of the first one. These two shows the positive energy status, and this one shows okay this is a negative energy status. That is why this is inducing and this is inhibiting. So these three enzymes, again I'm repeating, citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate, all the three enzymes are involved in the regulation of citric acid cycle. So with this we can conclude citric acid cycle. And so this is a relation between the citric acid cycle and the glyoxylate cycle. Uh, Citric acid cycle will produce NADH and FADH2 that will form ATP. The main purpose of the citric acid cycle is to produce energy. At the same time, glyoxylate cycle is a variant of citric acid cycle, and this variant will form oxaloacetate, and this oxaloacetate involved in the formation of glucose or synthesis of glucose, and further glucose to amino acid production or nucleotide production, or otherwise oxaloacetate to amino acid or nucleotide production. So why these two are very important? Because the citric acid cycle is comes under directly the central metabolic pathway, and because energy is main priority. And here the glyoxylate cycle is not present everywhere, and based on the condition, maybe glyoxysomes are rich in glyoxylate cycle, and this glyoxysomes will produce more amount of oxaloacetate, okay, from the glyoxylate cycle, and so this one will liberate glucose by gluconeogenesis, and glucose to amino acids and nucleotides will be formed. So coming to the glyoxylate cycle, this glyoxylate cycle is required by aerobic bacteria to grow on fatty acids and acetate. For example, a bacteria which need a special metabolic pathway, okay, to grow on fatty acids and acetate. The plants and protozoa also have this glyoxylate cycle, okay, however, it is absent in animals. So we don't have this glyoxysomes, and the glyoxysomes is present in plants, and especially in the bacteria, it is having the glyoxylate cycle, and there is no subcellular organelle is present in bacteria because as it is a prokaryote, and the component of glyoxylate cycle, and as a part of glyoxysome, it is also present in bacteria to metabolize fatty acids and acetate. The glyoxylate cycle resembles a citric acid cycle, except it bypasses the two decarboxylation reactions. You can see one by one what are the two bypasses. So till this reaction is okay, and this is isocitrate formation, and this isocitrate is converted into the first reaction, okay, this is catalyzed by a first reaction, that is isocitrate lyase. Lyase means breakdown. You can see here this isocitrate is this carbon, this after fourth carbon, okay, this bond is broken, and automatically this will form a molecule is called as Okay, this one is succinate, and remaining fragment, okay, 2 carbon fragment is called as glyoxylate. So glyoxylate, again, it will enter into the pathway by the conversion into malate by the fusion with acetyl coenzyme A. The acetyl coenzyme A with the glyoxylate forms malate, and malate to malate dehydrogenase to oxaloacetate, and acetyl coenzyme A combines with citrate. So why this is formed? As I told a little earlier, okay, that is the main thing is to utilize fatty acids and also acetate molecules rich in acetate. So this is very particular for the bacteria that will grow only in acetate molecule, and also acetate rich nutrients, or maybe the media, and also the fatty acid rich media. Okay, and one more thing, okay, this is also very present, okay, in plants. So during the germination process, this will form the fatty acids degraded to form this molecule, okay, that is succinate, and further succinate, okay, this is utilized for the further production of the metabolites like, okay, amino acids and also nucleotides. Second reaction, okay, this reaction is the fusion of acetyl coenzyme A, and coenzyme A is liberated, and this is called as malate synthase. So malate synthase utilizes oxaloacetate, this 2 carbon. This is the 2 carbon of isocitrate combined with 2 carbon of acetyl coenzyme A, and coenzyme A is liberated. This coenzyme A is liberated, and remaining is fragment is malate. So 2 carbon from glyoxylate and 2 carbon from acetyl coenzyme A forms 4 carbon of malate. So with this, we can conclude glyoxylate cycle, and again coming to the regulation of glyoxylate cycle. And this glyoxylate cycle is regulated by the partial inactivation of phosphorylation. In E. coli, the isocitrate dehydrogenase activity is partially inactivated by phosphorylation when cells are grown on acetate. Okay, that means isocitrate dehydrogenase is an enzyme present only in the TC cycle. If this isocitrate dehydrogenase enzyme is inactivated, that means it is uh, partially inactivated by the phosphorylation. And this phosphorylation, why it is phosphorylation, means if the acetate molecules are more, means these acetate molecules will facilitate phosphorylation of this enzyme. Acetate also induces the enzymes of glyoxylate cycle. Therefore, the presence of acetate, isocitrate lyase activity increases, while the isocitrate dehydrogenase is partially inactivated. If acetate is present, automatically isocitrate lyase activity is increases. You can see here, this is isocitrate lyase activity, and this is isocitrate dehydrogenase activity. Once acetate is present, this enzyme is blocked, and instead of that, this enzyme is activated. So this is called as isocitrate lyase number 1, and malate synthase is number 2. And the isocitrate lyase requires a high intracellular concentration of isocitrate. The isocitrate lyase requires a high concentration of um, isocitrate, the composition. That means if this is inhibited, for example, isocitrate dehydrogenase is inhibited, the accumulation of more amount of isocitrate. This more number of isocitrate will facilitate the binding of isocitrate to the enzyme isocitrate lyase. Usually, this enzyme isocitrate lyase will not receive isocitrate as normal. If the concentration exceeds the normal level, then only it will work on isocitrate and forms succinate and glyoxylate. So that is why it is mentioned here. The isocitrate lyase requires a high intracellular concentration of isocitrate. It is presumed that the partial inactivation of isocitrate dehydrogenase increases the concentration of isocitrate, and resulting in an increase in influx through glyoxylate cycle. So there is a very highly coordinated the mechanism. First of all, if the acetate molecule is present, that will partially inactivate uh, the isocitrate dehydrogenase enzyme. So, this isocitrate dehydrogenase enzyme is inactivated. That is why it is in the dashed line. Okay, that means this is not present in glyoxylate cycle. All the enzymes are blocked. That means this one is totally blocked. Due to that, this is accumulated. If more amount of isocitrate means This will be acted on the first enzyme, that is isocitrate lyase, and this will form succinate and glyoxylate. And glyoxylate further reacts with number 2, okay, that is malate synthase, and forms malate. That's all about the regulation of glyoxylate cycle, and we can stop at this.